Thanks everybody for coming today. So today, Merritt is going to give a talk and kind of start a discussion on single gene solutions to crop yields. And I think Jim Holland is going to be featured here, it looks like. And I am really excited for this type of discussion and for Merritt to kick us off with all of her ideas. So please take it away, Merritt.
Thanks, Sarah. So I'm going to be talking about a paper that we're working on, looking at the perils and promise of single gene solutions to crop yields. Extraordinary claims require extraordinary evidence. And Jim Holland gave a really similar talk a few weeks ago, and I figured that we could combine forces and then give a joint talk together to talk through some of these issues. So where did this all start? Near the end of July, Science published this paper looking at how they increased yield in rice and also in wheat by around 40 to 70%. On Twitter, I went through a lot of the issues that this paper covered, like testing their yields in non-commercially competitive lines, and also just kind of comparing their yields to the national yields, and showing that it was significantly lower. And it kind of went off, and it looks like a lot of people learned something and interacted with it. And so that was really cool. But almost like a month or a few weeks later, another paper came out in Science looking at this time increasing yield in soybean by around 33%. And, I went through a lot of the same steps again in this tweet thread, explaining how, the lines that they tested things in weren't commercially competitive. And how they didn't have enough environmental replication to truly test a lot of these effects. And so, this kind of pointed out that maybe we need like a longer form description of what a lot of these problems are outside of Twitter. And from that, I started kind of diving into the literature and finding a lot of these high impact journals consistently publishing on these 8 to 68% increases in yield. And they're over a wide number of crops in these journals. But I left a little screen capture of like an alt metric up here for one of these papers, because they get a lot of views and a lot of tweets about them and blogs written about them and YouTube videos describing them. And they also get press releases that are published to a wider audience, like the New York Times and the Economist and the New Yorker. And so, these papers have broad reach and impact. But one of the more problematic things is that most of these yield increases never translate to significant yield gains for major crops. And, one, it perpetuates the myth that these genes exist, but it also diverts time and funding and expertise away from proven plant breeding methods that actually work and deliver on yield gains. And this is genomic selection, and I'll talk about that a little bit later on. But current yield trends are really insufficient right now to meet growing demand, and they need to increase from 60 to 110% by 2050 to meet the projected growing demands of a growing population, climate change, and also diet shifts. And so messing up on yield has drastic consequences for feeding the planet. And so from a lot of these yield papers, I compiled 5 of the common problems that typically happen. And Jim's going to talk about the first 2, and I'll talk through the next 3. But essentially, they range from things like insufficient environmental and plot replication, like focusing on individual plant performance rather than plot yield. And that starts getting into looking at plant density and plant competition. Some misunderstanding of yields and different yield components, very limited testing of elite and diverse germplasm, and then some issues resulting from plant transformation and gene editing itself. So I'll let Jim go.
Right. So what I want to talk a little bit about was some of the problems are just not sampling the target environmental variation that represents where these crops are actually grown and need to produce yield. So breeders tend to test crop performance in relatively large samples of environments, and for 2 reasons. One is to get precision on the estimates of variety differences, and then also to increase the inference scope of when you're claiming you have a yield gain. Hopefully that corresponds to a reasonable sample of really the target environments. So again, just the difference between these things is precision is how much variation is there around that mean, and inference is like how widely we can interpret that mean. So this is some figure from a classic paper from Allard and Bradshaw. This is the year I was born, and I'm an old guy, so like this is this has been well known for a long time. But you know, breeders deal with these issues where the relative performance of two varieties, and here he shows these two varieties A and B as performance slopes against two environments, X and Y. And you know, it's sort of ideal to have this situation where those performance slopes are parallel, so that the difference between variety A and B doesn't really matter what environment you're in, it's very consistent. That's the case of no genotype by environment interaction. But the other cases drawn here are either like this case here in yellow, where the genotype by environment interaction differences are due to the ranking is the same, but the relative differences can vary among environments. And that's a bit of a problem, but the real problem comes in when you have the crossover interaction. So in environment X, one variety could be best, in environment Y, the other variety could be best. And this happens pretty much all the time. And this is why you need to sample a lot of environments to understand where one particular variety is doing better than another variety or a group of varieties. So I think we can go. Yeah, so by doing an experiment where you increase the number of replications in one environment is good for improving the precision of the yield differences in that particular environment, but all your inferences need to be restricted to the environment that was tested. And if you ask the question, what would happen in a different environment? For example, what would happen in the environment of a farmer under production? You actually have no idea. You have no basis for making any inference there. So that's a real problem. So you need to test things across environments, which, you know, allows you to at least understand, are these variety differences consistent across environments? If they have interactions, what is the restriction on the set of environments where one variety might be doing better than another? So we can go to the next slide. And again, I'm just focusing, you know, part of the reason I'm looking at these old literature examples is just to make it really clear that these principles have been understood for a very long time in the plant breeding communities. There's nothing new here. This should be well understood. I also focused on soybean examples here, just because, you know, I was presenting about a soybean paper, but you can find examples like this in essentially all crops. This example is looking at how much variation is due to genotype environment interaction compared to genotype main effects. And these were in these multi-state yield trials for soybeans. And so they break them into different maturity groups. And in each maturity group, they present how much variation is due to genotype main effects. And that's the part that's consistent across environments, and how much is due to this interaction of variety rankings or relative performances changing. And in all but one of these maturity groups, the yellow bar is the G by E variance component. That variance component is bigger, well, all but two of those cases, the G by E variance component is bigger than the genotype main effect. And in some cases, much, much bigger. And again, this is a soybean example. I just went back and I looked at our data from the Genomes to Fields project. And there, the genotype by environment interaction variance is considerably bigger than the genotype main effect variance. So it also happens in corn. And again, I think it pretty much happens in all crops that are grown across wide geographic ranges. So this is why plant breeders tend to want to sample a lot of environments before they're going to release a variety. And again, this is a soybean example, and this is public sector. I just want to make the point that even in the public sector, which is small scale compared to commercial sector, that the numbers of environments sampled before release is reasonable. And I would say typically it's something like at least 15 locations. And usually that's spread out over multiple years, because there's a lot of year-to-year variation is really important to sample. And most breeders won't release something as a variety unless it's been tested to that extent. So we can go to the next slide. Yeah. Okay, so you need to sample your environments really well. And then one other thing that seems like it, unless you've done these experiments before, it might not seem very important, but measuring yield on individual plants can get you into problems because the individual per plant yield responds to how densely you've planted the plots. And that, again, this is true more or less across all crops. And this is examples again from soybeans. But on the left, you can see as the planting density increases, you get this nonlinear increase in yield per area. And then if you take that and you convert it to yield per plant, you get a nonlinear increase the other way. And so the issue is if you've comparing two varieties and it just turns out that their planting densities are different, and that can happen just due to germination differences, or some plants died. Any of those differences are going to contribute to measurable yield differences per plant. So then if you then scale that up and claim I'm measuring yield per hectare, you are not really doing that. And you're not really comparing variety differences at that point. You're comparing differences in the response to planting density. So that's another big issue. And then I think and again, I think the corn people know this pretty well. This is classic stuff from Don Duvik. If you compare old versus new hybrids at high densities where they're grown typically, that's the kind of top curve. So over time, yields have gone up. And this is what we talk about all the time as corn breeders. We've done a great job increasing yield per hectare. Part of that's been because modern varieties can be planted at much higher densities. If you actually can, look at the individual plant yields over time, that hasn't changed at low densities. So if you look at that bottom slope, that's the low density planting. There's not been very much change in yield per plant over time. And so I think the converse is also true, that if you measure a difference in single plant yield, that may not at all translate to differences in yield per hectare once you scale that up to a proper farming and management system. And then I think maybe one more slide here. This is one more thing that people need to think about, is making sure you have plot sizes big enough, because in small plots, there's a lot of influence from having either a neighbor plot that could be competing with the plot you're trying to measure, or if you have big alleys and you have big borders, not having competition there causes you to have potentially overestimating the yields considerably. And this is a picture from a wheat experiment where they actually had multi-row plots of the same genotype, and then they measured the individual row yield and showed that in the middle of the plot, let's call this the typical, probably realistic estimate of yield here at 100%. As you go out toward the empty border, the closer and closer you get to that empty border, you can more than double the yield within the rows. So if you then are not using these large plots and you're not ignoring the outside rows, you're going to be heavily influenced by the lack of competition. So you need to worry. Basically, anything done in small plots is probably going to be unreliable. Am I done? Oh, yeah, I think maybe this. Yeah, one more slide. Yeah, and that's just one more dig on keeping up with the literature. This is actually a thesis from Kieselbach in Nebraska, and you can see the date there. This is 1917. You can go back through the old literature and you can see the technology at that time for planting was horses. And, so the technology was very old, but actually all of these ideas are laid out quite beautifully in this thesis. If you look, he's got a series of sort of thesis points at the beginning. It's a thesis, but he's got little summary points. So one of them is showing that interplot competition is a big issue. So if you're bordering plots, small versus tall plants, that's going to cause problems. And then he also lays out in this thesis that there's this nonlinear response to planting density, and that you need to evaluate things at different management practices, in particular planting densities, because you may find a yield advantage in one planting density that doesn't carry over to another. And then he also has this really important point on communication, which is when you do a yield trial, you need to give people enough information that they can actually evaluate. And I have a problem with a lot of these papers where they don't even communicate enough as to what they've done for you to evaluate what they're doing. So I'm done there. I will turn it back to Merritt. Thank you.
Okay, thanks, Jim. Okay. One of the next, common problems in these papers is a misunderstanding of how yields and different yield components behave. And so for a lot of the major field crops, yield is measured as the weight of dry grain or the dry matter content of the storage organs harvested per unit area. And what a lot of these papers tend to do is over-focus on individual components that can lead to really misleading yield estimates. And so what I mean by that is only measuring grain length or grain width. And so comparing an increase in grain width in a wild type versus a mutant and claiming that increase in width as an overall yield gain for the plant and not necessarily across the whole holistic system. And there's other examples of only measuring per plant yields or differences in grain fill in a wild type versus a mutant, having early flowering versus regular flowering, or biomass accumulation, which might not always increase overall yield for a plant. And there's also like issues with studying these effects in non-crop species like Arabidopsis. And, there's nothing wrong with studying each one of these things and how they do contribute to yield. It's when you solely study those things and focus, entire results and discussion sections around how your yield increase was slowly on a grain width increase that this starts to become a problem. And so true and accurate yield estimates have to involve, numerous genetic backgrounds screened across multiple environments under these farmer relevant management conditions across all yield components and not one in particular. Up next is limited testing of how these genes behave in elite and diverse germplasm. And typically the lines that are able to be transformed and used in a lot of molecular biology studies are not commercially competitive. And so a lot of these legacy varieties can yield a lot less than their commercially competitive counterparts. And this is an example within tobacco, looking at plot-wide yields of some commercially competitive lines from 2006, compared to a line that's able to be transformed called Petite Havana. And it yields around 1,600% less. And then, its commercially competitive counterpart. And Petite Havana is typically used to measure yield within molecular biology studies. So it's very off of what is available. And what kind of compounds this legacy variety issue is, anomalies resulting from the transformation or editing process. And so tissue culture itself can result in somoclonal variation. It's this heritable epigenetic variation that can lead to unintended effects on yield and other phenotypes. And this is solved only through backcrossing your transform line into a non-transform, hopefully commercially competitive line. And if you don't backcross these lines, it's really impossible to separate the genetic effects of like the transformation and editing process from the actual gene you're trying to test. And so it's a really important step in all of this that's usually missed. And so kind of switching gears a little bit and getting into like the population genetics and selection of these genes, any allele that has ubiquitously increased yield across environments and genetic conditions has probably already been fixed by selection, and specifically directional selection. And the only exception to this is any alleles with large effects in specific environments or novel environments, because these are unlikely to have been fixed across all populations. And they probably hold the best potential for stabilizing yields through plant adaptation and resilience. And so what are some examples of these yield stabilization genes? There is probably one of the famous ones for maize domestication, teosinte branch 1, that improves harvestability, taking this highly branched teosinte plant and providing a single stalked maize plant that's easy to go up to and harvest things from. There's teosinte bloom architecture that increases processing and consumption, kind of rolling back these hard fruit cases and allowing the kernels to be more easily edible. There's the BT genes in maize and other crops that help fight against insects and other, or just global warming induced pests. There's submergence 1 in rice that allows rice to tolerate flooding environments in those limited environments that do have flooding. And probably the most famous of them all, the reduced height 1 and reduced height 2 genes in wheat from the Green Revolution that allowed wheat to strive and thrive under high levels of nitrogen fertilizer and not lodge under or after windy conditions. And so I would argue that a lot of these yield stabilization genes only indirectly contribute to yield. They instead stabilize yield through changing the ease of harvestability and consumption, allowing plants to deal with specific abiotic or biotic stressors or responding to high levels of synthetic fertilizer. And so no single yield gene has been commercialized, and it's not from a lack of trying. And I have a little star next to this, and it's, as far as we can tell from public data, even though 2 have passed through regulatory steps, and I'll talk about those in a minute. But this is a great paper from Simmons et al. In 2021 in Plant Science. They essentially tried to document their 2-decade-long program at Corteva, where they tested over 100,000 total genes and for their effects on yield and yield stabilization. And a lot of these were just computationally, but they did end up testing around 1,600 genes actually within plants, or transformed into plants from 47 different species. And only 1.3% of all these genes, or 22 of them, show enough evidence to be validated. And the validation has to pass through like yield increases across, yield genes and also yield stabilization genes. And this is another figure from their paper. Most single gene constructs, once tested at scale, actually end up reducing yield. And so this is just a subset of their 145 constructs. Only 8 of them significantly contribute to yield and other quality traits. And most of these constructs are non-significant or significantly decrease yields. And this kind of highlights the importance of testing for genotype by environment, by management, by transgene effects, because even though these genes could provide some really great yield stabilization gene or yield gene, they might not necessarily be stable across environments. Okay, and now on to those 2 genes that have actually been approved. 78 total GM events have been approved, and they range across things from herbicide tolerance, insect resistance, and drought tolerance. But the 2 for yields, one is a transcription factor from Arabidopsis, it's from Bayer, and the other one is ZMM28 from Corteva. ZMM28 is a domestication transcription factor. And it only leads to a 1.6 to 3.9% increase in yields. And this is a figure from one of their papers looking at the increase in yield along the bottom and the event with ZMM28 minus the control without. And the line is no difference in yields, but essentially they tested this within 48 elite hybrids across 58 site year locations, and it only increased yield in like 80% of the environments they tested it in, not all of them. But these yield gains are very small and modest in comparison to what a lot of these high impact journals are reporting. They're 8 to 68% increases. The most promising genes actually have around 1 to 6% increases in yields. And so, yeah, the real promise of where these kind of yield genes and yield stabilization genes come in handy is with their incorporation with genomic prediction. And so what is genomic prediction? It is what breeding programs currently rely on in order to make genetic gain. And so in a typical breeding cycle, plants are planted out in the field in replicated trials and evaluated for disease prevalence, yields, and other quality traits. And at the end of the season, plant breeders select on the best individuals, cross those lines together, and then keep repeating this cycle until they get a variety that is stable enough to be released and commercialized. But with genomic prediction incorporates genome-wide markers for all individuals within a population to predict how well these genotypes will behave for these different traits in similar or novel environments. And genomic prediction has really allowed plant breeders to track, model, and select on the genetic effects of thousands of sites genome-wide simultaneously, and not only one gene at a time. And this drastically reduces the amount of time it takes to complete a breeding cycle. And so this is very anecdotal evidence, but very rarely are genomic prediction or genomic selection papers published in these high impact journals. And so I scraped this data from the Web of Science a few days ago, but I essentially searched for any papers across journals that mention genomic selection or genomic prediction. And typically they're confined into journals about animals, like Journal of Dairy Science, most popular for plant folks is probably G3, Frontiers in Plant Science and Crop Science. But after around 500 to 600 journals, Nature and Science come. And they only have 2 papers each, and they're not necessarily genomic prediction or selection papers within crops. One of them is in coral, and the other one is a like pangenome paper saying that they could potentially apply this to genomic prediction. And so it might be this lack of representation that could be why these single gene papers keep making their way into the spotlight rather than proven plant breeding methods that work, like genomic prediction and selection. And so from all of this, we kind of created a list of community guidelines to help researchers build robust experimental designs that don't get into these kinds of issues, but then also help readers and reviewers kind of critically examine this body of research. And they're all pretty basic things, but they start with using germplasm that is commercially competitive, backcrossing any transgenes or edits into multiple elite genetic backgrounds, testing all changes with sufficient environmental and plot replication to assess any genotype by environment, by management, by transgene effects, following crop-specific agronomic practices. And so, for example, if you are wanting to test the effects of a single maize or a single gene in maize on yields, for an application into like the US corn belts, you would probably want to fertilize under normal farmer practices, not till, probably not water these things, directly seed it into the ground and then mechanically harvest anything. It's not really realistic or comparable to compare those yields to something that you hand harvested, hand planted, and then treated very well with a lot of water and fertilizer that isn't necessarily comparable to what farmers are going to experience. Next is appropriate field designs to avoid any edge effects, any plant competition that might arise, and plant at industry standard density. Comparing to standards or negative controls, so compare your yield to what is present nationally or locally, and also kind of compare it to null constructs missing your gene of interest but are similarly backcrossed into an elite variety. Defining a minimum yield advantage. Some commercial companies require a significant advantage of like 2 to 5 bushels per acre in maize before actually going forward with any more validation. Prioritizing genes that evolution may have missed, and then also collaborate, because this is a lot of stuff and no one person knows how to do everything, but there are plant breeders out there that do this every year, year in and year out. And so a bunch of public sector yield testing frameworks exist already. One of them is the Genomes to Fields that regularly test maize varieties across the US every year. There's university-based breeding programs and experimental stations working on a wide number of crops. And then also internationally, there's the international variety trials through a lot of the crop innovation centers that do this for various number of crops. And so yield trials are affordable, but they are worth it. This is just a guesstimate on price. We estimate it to be a little bit lower than $20, but on a high range, if it's $20 a plot for 3 reps, 10 genotypes across 5 locations in 2 years, that's around $6,000. And yeah, inaccurate yield estimates have global consequences, and $6,000 for a multi-million-dollar project is kind of a drop in the bucket, in terms of price. And these yield estimates do have global consequences, not only for feeding the planet under climate change, but also in the investment of years of people's lives and years of their expertise spent towards chasing a lot of these single gene claims that don't ever pan out and don't ever produce results. And, there's also the allotment of research dollars towards these kinds of projects that don't lead to results. And this is especially more problematic in countries that need these yield increases, but suffer from publishing cultures that reward these high impact publications over incremental yield gains and product delivery. So, yeah. a comment that I got a lot on Twitter, and I've heard after other folks' talks on similar issues, is that a lot of these papers were just proof of concept experiments. They were really about other things, and yield was just, a part of it, not all of it. And, I would respond to that preemptively, that extraordinary claims require extraordinary evidence. It really becomes problematic when, the main focus of these papers shift into how their gene increased yields and how their summary abstract was about the majority of their yield increase. And then press releases start advertising on these yield increases that aren't actually realistic within plant breeding. And it's really only when yield increases are stable, repeatable, and show statistical support for the desired target environment and management scenarios should they then be officially declared and reported as yields increases. And so to kind of wrap things up, it's really working across the fields of molecular biology and plant physiology and plant breeding that we're really going to have the best opportunities to make meaningful gains in yields. And we can't forget that evolution has been the most persistent experimenter. It's had over 10,000 years to test the effects of all single genes within the genome. And, the real yield improvements moving forward will likely incorporate genomic prediction with the top 20 or 30 genes with really small incremental effects in yields, and then also yield stabilization genes. But, in a world facing climate change, hunger, and a need for massive carbon sequestration, there's no more accurate time to accurately measure and improve and deliver on yields to farmer fields where all of these results really, truly matter. And so with that, I would like to say thanks. This paper is hopefully coming to a journal near you all soon. I had the opportunity to work with a lot of really great public sector plant breeders across a wide number of crops and countries to kind of assemble a lot of these issues and then community standards. And I am personally funded off a USDA NIFA predoctoral fellowship, and that I'd be happy to take questions. I kind of hope for this to be more of a roundtable discussion than a Q&A, but either works for me.